All right, so to look at what happens next, what we want to do is we want to look at figure 12.9 in your textbook. This is on page 384 of your text. I'm not going to draw all these out. I'm going to save you the torture of this. I also have this figure posted on my website from an earlier edition of your textbook as well, showing these same basic steps. So you have a couple of different places where you can find this figure, right? So before we showed this figure, I had a hand-drawn figure showing you how troponin, tropomyosin, myosin, and actin all interacted with one another. And what we said is that myosin can't interact with actin when calcium's not present because we have tropomyosin blocking that binding site. And that's absolutely true, all right? It's not to say that they can't interact at all, they just can't interact strongly enough for the muscle to contract. So as it turns out, what we have typically is we have myosin being very weakly bound to actin in a relaxed muscle, all right? And that's what we have starting right down here is we have myosin being bound very weakly to this relaxed actin. So this muscle is relaxed at this point, all right? Now, in order for this to occur, we have a couple of binding sites, and you can see those binding sites right here. And on this binding site, we have an ADP, adenosine diphosphate, and we have an inorganic phosphate. This is an ATP that's been hydrolyzed. We're going to get to the significance of that here in just a second, but for now, just know that we have myosin weakly bound to actin. This muscle is relaxed, and we have this hydrolyzed ATP that's sitting here in these binding sites. Okay. And now we have that calcium signal coming in. Okay, this is that calcium that's been released from the sarcoplasmic reticulum. It's going to bind to troponin, and it's going to move tropomyosin off of the binding site. Okay. When that happens, all right, we see a much tighter binding here. All right, we see a much tighter binding between the myosin and the actin. And we also see that inorganic phosphate getting kicked off of the binding site, right? That's been being released from the myosin head. When that happens, the myosin head swivels, okay? It literally does this. It moves. It literally swivels. And that is called the power stroke. And it moves the actin towards the M line of the sarcomere, all right? When the ADP releases, that marks the end of the power stroke, right? So the myosin releases the inorganic phosphate, and we see the swiveling of the head, okay? And that pulls the actin towards the M line, okay? Pulls the actin towards the M line. And then the ADP is released. So the ADP pops off of the myosin, all right? Now, in this state, the myosin is still going to be very strongly bound to actin. These two molecules have a strong affinity for one another, and it causes this strong binding to occur. This myosin will only release from the actin if another ATP molecule comes and binds to it. And that's what we see happening here. ATP binds and it's released. I think some people sometimes think that the ATP is required for the power stroke, and I guess in sort of a roundabout way it is, but why we really need ATP for muscle contractions is we need it to release myosin from actin. If we had ATP absent but calcium present, what we would be is we'd be in a state called the rigor state. You might have heard of rigor mortis, which is where the muscles get really stiff right after death. Well, what's happening is that there's no ATP because you're dead, and there is calcium present because the sarcoplasmic reticulum is broken down, and that causes this rigor state to occur. The muscles get very stiff for a few hours after death. All right. Of course, after a couple of hours, all of the protein breaks down, and then the muscles get sort of flaccid again. But for a few hours, we end up in this rigor state, and this happens very briefly during the process of a muscle contraction. So ATP binds to this binding site on myosin and causes it to release. All right. The ATP is hydrolyzed again. Okay, there's an enzyme called myosin ATPase that causes that hydrolysis to occur. And then we start the process over again. If the muscle is continuously being stimulated, we have a constant calcium signal, and we have another power stroke that occurs, right? It releases, another power stroke occurs, and the cycle continues, and the myosin heads walk that actin towards the midline. When that happens in enough sarcomeres, it causes the muscle to shorten. All right? There's a really good description of these steps in your book. Feel free to rewatch this as many times as you need to. This is a really important series of steps, but it's a really cool series of steps as well. All right. Remember that there are enough myosin heads present, right? They're only showing 2 here, but there would be many, many more than that. That what happens is that they're all sort of walking at once, and that just creates a nice smooth movement of the actin towards the M line. Okay, so with that in mind, move this book out of the way. What happens when the contraction stops, right? That's going to essentially happen for as long as there's a calcium signal. But eventually, the muscle is not going to be stimulated anymore. And what's going to happen when that calcium signal stops, when we stop stimulating the muscle? Well, everything that we've been discussing essentially is going to reverse itself. But there are a couple of key points that I want to point out about muscle relaxation. Okay, what do we have to do to relax the muscle? All right, a couple of things that I want you to think very specifically about in this situation. The first one is that we've got to get rid of acetylcholine. As long as acetylcholine is bound to the muscle cell membrane, the muscle is essentially going to be stimulated. All right. So there's an enzyme called acetylcholine esterase. Acetylcholine esterase. We typically abbreviate that as just ACHE. Okay, is going to remove acetylcholine from its binding site. Right? It removes acetylcholine from receptors. All right. The next thing that we have to do, that we have to consider for muscle relaxation, is we have to consider getting the calcium signal out of the cell, right? Remember that calcium is allowing actin and myosin to bind with each other. We have to stop that process. So we have to reclaim calcium into the sarcoplasmic reticulum. Okay, we have to move the calcium from the sarcoplasm back into the sarcoplasmic reticulum. There are pumps on those ryanodine receptor channels that allow that calcium to get pumped back into the sarcoplasmic reticulum. What we're doing here is we're moving calcium from an area we're moving calcium against a concentration gradient. Let me say it that way. We're moving calcium against a concentration gradient, and this requires ATP. Okay, this requires ATP. This is an active process. Similarly, what we're going to have to do at the level of the acetylcholine receptor channels is we're going to have to be able to move sodium and potassium back into their correct spots as well, and that's also an active process. Actively pumping sodium out of the cell and pumping potassium back into the cell is also an active process. All right? But these two things, right? These two things are critical for us to relax the muscle after it's been stimulated. We have to stop the signal at the level of the cell membrane, and then we have to reclaim that calcium back into the sarcoplasmic reticulum. Both of those, to some extent, are active processes, so they require ATP. And I think that sometimes we think about muscle relaxation as being passive, but it is indeed an active process. So, and all of this is said and done, what are we left with? Let's take a look at this sort of graphically. What I'm going to do is I'm going to draw a divider right here, and I want to draw a graph, and I want to talk about muscle tension on the y-axis versus time on the x-axis. What we're going to do is we're going to take a look at this. So we have a relaxed muscle, right? This relaxed muscle is sitting here like this, a little bit of tension. We'll talk about that in a little while. And then what we're going to do is we're going to stimulate it, right? The stimulus is going to be a pink arrow. Okay? It's going to be a pink arrow that looks like this, right? So we're going to stimulate the muscle right here. Okay, we're going to stimulate the muscle right there. Now, do you think that the muscle is going to contract right away? In other words, do you think that we're going to see an immediate rise in tension once we stimulate the muscle? It turns out that everything that we've talked about does take a little bit of time. So the muscle is not going to rise in tension right away. There's going to be a brief period of time where nothing is really visible in terms of increased tension in the muscle. But then we will see a rise in muscle tension as the steps that we've discussed sort of take place. Right? We see the release of acetylcholine from the muscles, from the neuronal cell. We see the binding of acetylcholine, the movement of sodium and potassium. We see the depolarization of the muscle cell, the release of calcium from the sarcoplasmic reticulum, so on and so forth. All of that takes a little bit of time. But then the muscle is going to gain tension. Okay? Now, I've only stimulated it once, so what's going to happen is the muscle is going to gain tension, and then those steps of relaxation are going to occur, and the muscle is going to lose tension. So we end up with something that looks like this. All right? This is called a muscle twitch. Okay? The twitch can be very clearly divided into a contraction phase, right, and a relaxation phase. All right? So there's two phases that we have in this muscle twitch. This muscle twitch, I have not put a time on this x-axis other than to say that it is time, because it does vary in speed. Okay, very roughly speaking, muscle twitch has a range between about 50 and 200 milliseconds. Okay, we're looking at about 50 to 200 milliseconds. Different muscle fibers in our body do differ in their twitch speed. Some have a relatively fast twitch, some have a relatively slow twitch. We're going to talk about slow twitch versus fast twitch muscle fibers here in just a little while. But that's what we have. This is a muscle twitch. Everything that we've just described. I think I'm getting close to an hour and a half of going through this with you guys, and all of the events in our muscles happen in way less than a second. So if only I could talk that fast, right? Okay, so now that we understand what's happening in a single muscle cell with a single stimulus, let's look at a muscle by itself, right? So muscle cells don't normally function in our everyday lives by themselves, right? We're using whole muscles to do tasks, whether it's going to the gym and lifting weights, or whether you're making a sandwich or walking across campus, you're talking about whole muscle function. So what's going on with whole muscle function when we do particular tasks? And that's what we want to look at next. I think probably the most noteworthy observation that I make about my muscles is the fact that there are some times when I need to have a really strong contraction, and there are times when I don't need as strong of a contraction. And our muscles have this remarkable ability to generate strong contractions or weak contractions, or somewhere in the middle. All right? We call those graded contractions. Okay? We call those graded contractions. How do we generate graded contractions in our muscles? In other words, how do we generate strong contractions, and how do we generate weak contractions? Now, there's two ways of looking at this, and there's two ways that we do it. All right? The first way, the first one I want to talk to you about, is increasing the frequency of stimulation in a motor unit. Increasing the frequency of stimulation in a motor unit. So let's go ahead and graph that. All right, see what that sort of looks like. I'm going to describe this as I go. It's the same graph I had last time. We're going to have tension on the y-axis, and we're going to have time here on the x-axis. Again, I'm going to use pink arrows to indicate a stimulation, and I'm going to use green as my trace here. So here I have a relaxed muscle cell. There's no tension on it, and I'm going to stimulate it just the same as I did before. So there's my first stimulation. And you'll see that there's this latent period, and then we see this sort of muscle twitch occurring. All right? Good? Let's go ahead and stimulate it again. So we have muscle being stimulated again, and we see the same thing happening. All right? Muscle twitch, about the same tension occurs. But here's the thing. What I'm going to do now is, before I can come back to relaxation, I'm going to stimulate it again. Right? So here, these stimuli were some distance apart. You'll notice these two are closer. I've increased the frequency of the stimulation. And what we see from the muscle is we see that it responds again, but it responds with more tension. We have a stronger response. This guy comes back to resting, and we have something that looks like that. All right? What I did here is I took one contraction and I added a second one onto it, right? So we have this sort of delta right here, this sort of difference in tension because I increased the frequency of stimulation, all right? What I've essentially done is I've added two muscle contractions together. When I add two things together, what do we call that? We call it summation. All right, we call that summation. And that's what's occurring here. Let's take it one step further and go even more. All right? So I'm going to stimulate it here. We're going to see, I'm going to run out of room on my graph if I'm not careful here. We're going to see our stimulus, okay? And now what I'm going to do is I'm going to draw my stimuli in a little bit more here in just a minute. I'm going to stimulate it again and again. I'm going to end up with something that looks like that, right? So my stimuli are here, 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 so on and so forth, all right? Here what I'm doing is I'm stimulating it enough that I eventually reach kind of a max tension, right? For that particular muscle cell or group of muscle cells, I've reached a maximum tension. I can't go any higher than that. And you'll see that I kind of, even though I'm at this max tension, there is just a little bit of relaxation in between each one of those. Okay? We call this incomplete tetanus. Okay, we call that incomplete tetanus. In your everyday life, you aren't really ever going to encounter this. This can be encountered or caused with a muscle stimulator by somebody who knows what they're doing, but this isn't something that you really have a lot of experience with in your everyday life. It's more of a theoretical state than an actual state. However, we can take this one step further, and I'm just going to expand my graph here just a little bit. We can take this one step further. So this guy, I'm not going to stimulate it anymore. I'm going to let it return to sort of normal here. So here we are returning to normal. And what I'm going to do now is you'll notice that the stimuli were really close together right here. I'm going to move them even closer together. All right. So I'm going to really start to just hit this guy with action potentials. I'm going to hit this muscle cell with action potentials. We're going to see a rise in tension. We're going to get to that same max tension, but there's going to be no relaxation between stimuli. So we're going to receive this constant, smooth state of contraction. This is called complete tetanus. Okay? Called complete tetanus. And I'm not going to really go into detail drawing my stimuli in here. Just know that those stimuli are very, very, very close together. Right? So we're stimulating, 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 right? Keep these going just a little while further. Okay, so we're stimulating, 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 stimulating. And we see this smooth contraction that occurs, right? So when you hold a glass of water in your hand, for instance, and your biceps muscle is contracted, holding your elbow at a right angle, what you have is you're holding that biceps in a state of complete tetanus. That muscle is in a constant state of contraction, right? We often associate tetanus with the tetanus toxin, which you can get when you cut yourself on something rusty or something like that. And that does cause muscles to go into tetanus. But tetanus itself is a normal physiological function that happens in our everyday lives that helps us to hold things in place. And that's what we see happening here, all right? So for this group of muscle cells that we're stimulating in this motor unit, we can have complete tetanus being held at some maximum tension. Now, I drew these stimuli going past my green line, right? What's going to happen is if I continue to stimulate skeletal muscle, regardless of the muscle, if I continue to stimulate it, eventually the tension is going to fall off. You'll notice that in this space, tension is reducing, but there are still stimuli occurring. This is called fatigue. And skeletal muscle is somewhat unique about the muscle types in that it will fatigue with time. All right, muscle cells stop working. There are a lot of causes for fatigue. Your book has a really nice discussion of it. Know, though, that there are physiological factors contributing to fatigue, such as ion loss, like calcium. And there are also psychological factors. That simple fact of you saying, oh, I can't do it anymore, right? Whether you're lifting weights or holding something or something like that. There are different factors that contribute to fatigue. Once a muscle cell fatigues, it has to rest before it can continue to do work again. All right? And that sort of leads us to the next thing dealing with graded contractions, right? So here we see what happens when we increase the frequency of stimulation in a motor unit. We see that we can go from a relatively weak twitch to a muscle fiber with more tension. There's a maximum tension, but we can go to a muscle fiber that contracts with more tension simply by increasing the frequency of stimulation. Now, how can we do this if we want to lift something heavier? Remember that a muscle is made up of thousands of muscle cells, often controlled by many, many motor units. So we've only talked at this point about one motor unit. But what happens if we want to contract a muscle cell that is, or we want to contract a muscle with even more force to lift something even heavier? Well, if we want to do that, we have to use more motor units. Okay? We'll add to our list right here and we'll say increase number of motor units stimulated. Okay, this is called motor unit recruitment. Okay, so let's say that you're holding something for me and you're holding a book, right? And I turn around with another book and I say, oh, hold this one as well, and I put it onto your hands, right? Now, all of a sudden, your load has become heavier, all right? And the tension that you are generating is not enough to support that load. So what do? You are going to stimulate more motor units, all right? Your brain says, uh-oh, this load is heavier, I need to do something. So what it does is it activates another motor neuron. And that motor neuron innervates multiple muscle cells and it causes more cells to contract, generating more tension. All right, I load another book on, you activate more motor units. Okay, another book, more motor units, so on and so forth. And that process sort of continues for as long as you have motor units until you reach the maximum tension of the muscle. All right. This is a really cool process, right? You're increasing the number of motor units that you're stimulating, all right? Essentially what you're doing is, as a load gets heavier, you're calling in reinforcements from more muscle cells. So if you're holding a book for me that's relatively light, not all of the muscles in your biceps, for instance, are being stimulated. Maybe only a fraction of them are. But as the load gets heavier and heavier, you're using a higher and higher and higher percentage of those muscle fibers in your biceps to hold that load in place. Okay? Now, keep in mind that I said that these muscle fibers are eventually going to fatigue, right? So if you're holding a relatively light book for me, your elbow is contracted or flexed, your biceps is contracted, what's going to happen is that those muscle fibers that you're holding that book for are going to fatigue with time. Well, what's going to happen is that you can essentially sub players in off the bench if that happens, right? The muscle fibers that you're using are going to fatigue. What you can do is you can bring in more muscle fibers as needed, right? Let the fatigued ones rest and use new muscle fibers to hold the book in place. When those fatigue, you use new motor units, and so on and so forth, and you kind of cycle through, all right? Now, if you're holding something heavy for me, you know that you're not going to be able to hold it for as long, right? What's happening is that more of your players are out on the court, so as you call in other players off the bench, so to speak, to sub in, you have fewer fresh muscle fibers, and as a result of that, all of them fatigue sooner, and you aren't able to hold the book or whatever it is for as long. All right? So we have a number of different ways in which we can achieve graded contractions in skeletal muscle. Okay, so moving on, what we want to do here is we want to look at a couple of other sort of assorted things dealing with skeletal muscle. The first thing that I want to look at is the fact that I drew this graph specifically to where there's some tension on this muscle even when it's relaxed. That's called muscle tone. All right, so let's go ahead and take a look at muscle tone. We're going to look at isometric and isotonic contractions in this next unit, and we're also going to look at slow versus fast twitch muscle fibers.